The title of the poster is Challenges in Using Contrast Sensitivity and Reading Acuity as Dementia Biomarkers, the Effect of Variability. The authors are Walter Wittich and Natalie Phillips. The affiliation for Dr. Wittich is the Université de Montréal at the School of Optometry. The affiliation for Dr. Phillips is the Department of Psychology at Concordia University. Both are in Montreal, Quebec, Canada. This is a poster presentation at Vision 2025, the 15th International Conference of the International Society for Low Vision Research and Rehabilitation, that takes place from September 8th to 12th, 2025, at the Palazzo dei Congressi in Florence, Italy. Introduction. Changes in visual function, such as acuity or contrast sensitivity, have repeatedly been demonstrated among individuals living with dementia, such as Alzheimer's disease or mild cognitive impairment. However, few studies measured both these visual functions in the same cohort, and many diagnostic subgroups have not been examined. The purpose of the present study was to explore reading acuity and contrast sensitivity in older adults across 10 diagnostic groups compared to healthy age-matched controls. Methods: we analyzed the data of the COMPASS ND cohort study with focus on binocular reading acuity measured by the MN read and contrast sensitivity measured by the Mars chart using habitual correction. Both measures were taken under standard room lighting conditions. Participants were recruited from memory clinics, and all underwent professional assessments to determine clinical grade diagnoses. They were then categorized as healthy, in equals 161, subjective cognitive impairment, in equals 138, mild cognitive impairments, in equals 261, mild cognitive impairments with vascular contribution, in equals 153, Parkinson's with mild cognitive impairments, in equals 39, Parkinson's with motor symptoms only, n equals 0, 79, Parkinson's with dementia, n 15, frontotemporal dementia, n 30, Lewy body dementia, n 128, Alzheimer's disease, n 104, and Alzheimer's disease with vascular dementia, n equals 75. The presence of established ocular diagnoses was based on self-report data. For each group, we plotted reading acuity on the x-axis as a function of contrast sensitivity on the y-axis and calculated 95% confidence ellipses. We used the ellipse circumference as well as horizontal and vertical ellipse diameter as measures of variability and data dispersal. Results: ellipse models for 95% confidence captured between 90 to 97% of data points across the respective diagnostic groups. Compared to healthy controls, data dispersal, ellipse circumference, was largest in frontotemporal and Lewy body dementia, followed by Alzheimer's disease with vascular dementia. Horizontal ellipse diameter, reading acuity, was largest in frontotemporal dementia, whereas vertical ellipse diameter, contrast sensitivity, was largest in mild cognitive impairments with vascular contribution and Lewy body dementia. The Mars letter contrast sensitivity test features 48 letters, with each letter being a 0.04 log contrast sensitivity unit step. The MN read acuity chart typically contains 19 sentences or lines printed at varying print sizes, ranging from plus 1.3 logmar to minus 0.5 logmar and 0.1 log steps. Therefore, 0.1 logmar units are the equivalent of one line of 5 letters. Reading acuity ranged from 4 to 11 chart lines with the respective diagnostic groups. Contrast sensitivity ranged within 1 to 2 chart lines within the respective diagnostic groups. Tilt is relatively stable across diagnostic groups, indicating independence of the two vision measures. The percentage of data captured by the respective ellipses is high, ranging from 0.90 to 0.97. Reading acuity is the lowest in the Parkinson's dementia, frontotemporal dementia, Lewy body dementia, and Alzheimer's disease with vascular dementia groups. Contrast sensitivity is the lowest in the Parkinson's dementia, the Lewy body dementia, and the Alzheimer's disease with vascular dementia groups. Variability is the highest in the mild cognitive impairments with vascular contribution, the frontotemporal dementia, the Lewy body dementia, and the Alzheimer's disease with vascular dementia groups. The proportion of participants with self-reported vision diagnostics ranged up to 23.9%, but did not correspond well with impairments. Conclusion: data variability of visual function measures among individuals living with different dementia types is generally highly heterogeneous. However, this effect appears to be more pronounced in some diagnostic groups. The use of reading acuity and contrast sensitivity as potential behavioral biomarkers for dementia therefore remains limited. Self-reported diagnostic information can range widely across severity, which may partially explain why pattern of outliers did not appear to be explained with this information. In this sample, vision may play a more important role during assessment in the accessibility of cognitive testing materials.